All right, I'm here. Is everybody else here? Getting ready for this class. Because we're going to be doing some hands-on stuff. I'm live streaming with the cell phone today. Got all the materials I need, right? All right. So, I'm Josiah Zahner, and this class over the next 4 weeks, Mondays at 4 p.m. Pacific time, we are going to be going over how to grow and engineer human cells. Now, if you have not signed up for the class, check it out at the-odin.com. We send you everything you need. If you're just here to learn and you plan on buying the kit at a later time or messing around, that's great also. So we sent out the kits. You should be receiving them shortly. We wanted them to arrive around the time or after the time of this class, so you have an idea of what to do actually when the human cells arrive. The human cells are a little bit more finicky than normal cells, than like bacteria or yeast cells that you may have worked with, or even plant cells. So just treat them with respect, right? That's what I like to say. Don't expose them to a lot of changes in temperature. Try to keep them as sterile as possible, but just generally treat them with as much respect as you can. Now, human cells don't really like lower temperatures. They can handle room temperature pretty okay, but they prefer a temperature near 37 degrees Celsius. So that's why we included a heat mat in the kit. Now the heat mat has a temperature controlled thermostat, so it'll keep around 37 degrees. Now, because if you put stuff directly on the heat mat, it will be exposed to outside air and all the stuff floating around, we suggest you put it inside a Tupperware container. This helps with protecting it from, all the stuff that's going around in the air. And it'll just keep it more sterile. It's totally possible to do it without doing that, but it's just, it helps you out. Helps you out a little bit. Pipettes. If you've never used a pipette before, make sure that you learn how to use a pipette. We're going to be using them in this class. I'll go over it briefly. Pipettes. This is the plunger. You have to think of it like a turkey baster. A turkey baster, you squeeze, then you suck up the liquid. A pipette's the same way, except instead of squeezing, you push down. Now, when you get your pipette, you'll see that there's two stopping points. There's the first stopping point and then the second stopping point. The first stopping point is what you want to use to draw up liquid, and the second stopping point is what you want to use to expel the liquid, right? When you turn the top, that's how you change the volume that you're going to draw up. So know what volume pipette you're using, and then that'll let you know what volume you are drawing up, right? So this is 100 to 1,000 microliter pipette, and that means that the numbers here are, in the hundreds to 1,000 microliter. The back of the pipette, the buttons on the back, that helps you expel the pipette tips. So if you got a pipette tip like this, after you finish using it, eject it. Don't reuse pipette tips, especially when we're working with human cells. We want to keep everything as sterile as possible. So don't reuse your pipette tips. Eject them, throw them away, put them in bleach to kill any cells or bacteria on them, but don't reuse them. All right. Now, I guess we can start here. So we're going to work on my wonderful lab bench today. The first thing you want to do, clean it down when you're working with human cells. If you don't have any isopropyl alcohol, just order some off of Amazon. Get some isopropyl alcohol. You're going to use it a lot, but these Clorox wipes are also great. Just clean down your work area. You know, you're trying to make sure that everything you do is as productive and successful as possible. So, just clean everything down. It's good to have whenever you start doing your experiments. Whenever you pull out your human cells, just clean down your kitchen table or whatever, kitchen counter, whatever you're working on. Just so it's it's not going to be sterile, but it's going to be fairly clean. The number one problem you're going to run into is bacterial contamination. Probably. So you want to try to be as clean, as sterile as possible. Same thing with your hands. Whenever you're working with the human cells, always wear gloves if you can. We sent a bunch. You can reuse gloves. It doesn't matter. You're just trying to keep the bacteria from your body and your hands outside the culture. It's a good idea. You got some isopropyl alcohol, put a little bit on your hands, rub it all over your gloves, clean it all up. If you even want to get, a little more clean, put it on a paper towel, clean off your forearms, all that bacteria and dead skin and, fungus and all that stuff. Clean it off. And that'll help your future experiments totally. All right. Dave, it doesn't really matter. Some people say that 70% alcohol works better than 99% alcohol, technically. We use 90+% isopropyl alcohol for other stuff, so it's generally what we keep around. I don't think it matters too much. Whatever works for you, whatever scientific phenomenon you believe in. Yeah, I think I read somewhere or something that maybe 70% works better at sterilization than 90+%, but who knows? All right. So what you'll receive in the mail is a tiny flask that looks like this. Now your flask will be all full of a red liquid, and that's media. Now, if that media looks really cloudy, contact me immediately. That means that it got contaminated. We'll just send you some more. If it looks relatively clear, then and pinkish, then you're pretty okay. So what you want to do when you receive your flask is you want to dump off a bunch of the liquid until maybe you have a lot less, right? So maybe like that much, maybe a little bit more. So the media will probably be pinkish in color. So this media that we're using, it actually changes color based on the pH. So when the media starts to get, more orangish, here's the normal color of the media. When it starts to get more orangish, then that means that the pH is changing. There's a lot of cells growing, and usually that means it's time to either use your cells or replace the media, right? So, it has a color indication pH dye. So, use keep track of that. Now, an easy way to tell if you have cells growing or your cells are alive, if you take your flask and you hold it up to the light, let's see if we can get this in focus. There we go. You can see a bunch of little dots. Like the flask looks opaque and it looks like there's stuff growing on it. Right? On the bottom. So the flasks have a top and a bottom, and it should be pretty obvious what the top and the bottom is. But on the bottom of the flasks where all the cells grow, you should be able to see the opaqueness as little dots. There's pictures in the tutorials. I'm trying to get it on camera, but it's hard. It doesn't want to focus. What happens if I put down the camera and go like this? See if I can focus on it. Yeah, you could kind of see it there. You see how you see all those little dots? Those are the little colonies of the human cells growing. So if you hold up your flask to light and you don't see anything like that growing on there, it's not a good sign. It means your cells are dead, right? So generally, when we're working with these cells, HEK293 cells, when they're living and they're healthy, they are attached to the vessel. When they're not healthy, they're not attached. They'll just float off and die. Now, this plastic is special plastic. Generally, cells do not want to attach to plastic. So you can't just grow cells on any plastic. They use certain chemicals to modify the plastic because plastic is very hydrophobic, right? Plastic repels water, it repels liquids. It doesn't like things to bind to it. So they modify it, and they usually call this tissue culture treated. So if you're looking to buy different, human cell culture supplies, and you're looking on like eBay or Amazon or someplace like that, look for tissue culture treated flasks or tissue culture treated plates. Make sure they're tissue culture treated, or it's most likely that your cells won't attach. Now you can grow cells in what they call suspension culture, which is non-attached cells, but we won't really get into that in this class, and it's probably not something you'll really get into. But maybe you'll it's something you'll do in the future. So the cells we are using in this class are HEK293 cells, human embryonic kidney cells. These cells have been modified to be immortal. And it kind of means what you think it means, but not really. So generally, if you were to take cells from your body and grow them up, they're probably only going to survive like, I think it's like 20 to 30 passages, we call it, like replications, kind of. And this is because a lot of different things. They're normal, actual human cells. Their telomeres shorten, and they get old, and the cells die. So usually when you're working with human cells, you're working with immortalized human cells. This is what HeLa cells are, if you've heard of HeLa cells. And this is HEK293 cells. So HeLa cells and HEK293 cells are probably two of the most popular types of cells used in science experiments by scientists. And the HEK293 cells we found were a little more robust. They were easier to keep around and help survive. So we decided to go with the HEK293s and not the HeLa cells. HeLa cells are a little bit more finicky. So yeah, HEK293 you're going to receive. And from these cultures, you can propagate them indefinitely. But it's good to replenish your supply after a certain amount of time. Now you can store human cells frozen, but you actually have to store them at really cold temperatures in liquid nitrogen. So it is a fairly resource intensive process. Um, the other thing you can actually do is just keep your cells alive, right? So you just, every time that you split them or passage them, and that's just a process of stripping them off the culture vessel and reseeding them so they grow back up. Um, the cells can undergo mutations and other things. So, if you're using your cells a lot and you're splitting them and passaging them a lot, it's probably a good idea to just go to the source that you're buying them from and buy some more. The cost isn't that expensive. A few $100 you could probably find HEK 293 cells for online every 6 months. That's not too bad. So it's a good way just to make sure your cells are healthy, wealthy, and wise, right? Okay. HEK 293. Do HEK 293 cells have telomerase upregulated? Yes, I think they do. Minus 20C is not enough. So I did a bunch of experiments trying to figure out how best to store these cells. And you can store them at room temperature for a little while, like, 2 weeks or so, 1 to 2 weeks. But generally, the way to store them is just to keep them growing. Keep them growing and split them every week. They'll last you a while. If you want to go to the point of, 25 weeks, that's 25 splittings or passages. That's half a year. That's definitely a good point then to, get new cells. And that's not too bad, right? Cells last you half a year. That's a pretty good deal. So, you just keep them alive. Keep them going. Jewel, it kind of looks like a fingerprint. It more just looks like, there's something there. If you look at like the sides of the flask, they're really clear, right? Sides of the flask are really clear. But if you look at the bottom, it should be pretty opaque. And that's all those little dots are cells. Yeah. All right. So with your kit, you should get a microscope. If you have a CO2 incubator. So so here's the thing. So the media that we're using in this class, if you have your own media, you could always do whatever you want. But let me talk a bit about the media we're using in the class. So the media we're using is called L15 media. Now generally, people use DMEM media. Now DMEM media has sodium bicarbonate in it. It helps buffer the media against CO2, right? So CO2 enters the media. It provides CO2 to the cells, but none of this is needed. So if you use the L15 media in a CO2 environment, it would probably kill the cells. I don't know on how fast or on what scale, but the media that we're using does not require CO2. So you can grow the cells without CO2. So normally when you grow human cells, people grow them in a CO2 incubator. But yeah, you don't have to. Honestly, I don't know why. There are certain cells that are super finicky, primary cells, that you probably need CO2, but like L15 media works great. You don't need CO2 at all, and it'd probably be bad if you did use it, so don't use it. Now, in this case, we're also using FBS, fetal bovine serum, in the media. You don't have to use FBS. There's cheaper alternatives, but I wanted to make sure this class worked. So I wanted to make sure that your cells are healthy. So we're going to use FBS in this class. So FBS, fetal bovine serum, is basically the non-red blood cell part of a fetal cow. So fetal cow, when they take it out, when it's aborted or when it's unused, what they do is they will extract all the blood from it, and then from that blood they'll extract the serum, and then they'll sell it to scientists, because the fetal blood doesn't have a lot of antibodies, and so it's really, and it has all the nutrients, all the growth factors, a lot of stuff cells need to survive. There's also things that are like newborn calf serum, which is less expensive, where they just take the serum from a newborn calf. They don't have to kill it then. And, well, actually, I don't know if they kill it or not. I like to imagine that they don't. It's just like donating plasma or something, you know, when you were in college. But newborn calf serum, I've tested it. It also works. The cells grow a little slower. They're not as healthy. It's much, much cheaper than fetal bovine serum, FBS. But, you know, for this class, we're using FBS. If you want to use something else, there's a couple things you can use. There's Fetal Grow, which is a non-serum based serum alternative, or whatever you want to call it. It's about $138 per 500 ml. So generally, when you're doing cell culture, you use 10% serum in your media. So 500 ml would probably last most people like a lifetime. I mean, most biohackers. Because 500 ml is good enough for 5 liters worth of media. And yeah, it takes a long time to use 5 liters worth of media. So yeah, you know, Fetal Grow is good if you want to use that. If you want to go cheap, you know, newborn calf serum will also work. So is L15, 10% serum, which is, our serum is fetal bovine serum, and then antibiotics. So the antibiotics that we are using are gentamicin, streptomycin, and ampicillin. Now normally people use penicillin and streptomycin because our whole environment is not as sterile. Gentamicin is something that also people use, and I just like to throw it in there, give it the triple threat, and it works really well. It's rare when I get a contaminated culture. So the antibiotics shouldn't affect the cells. So you should mostly be okay. The antibiotics we're using shouldn't affect human or mammalian cells at all. But that's the media that we're using. L15 is really cheap. I think you can get it for like, $20 for 500 milliliters. Cost of shipping increases that a little bit. So you're talking like maybe $25 to $30 per milliliter. But, you know, it's pretty good. 500 mLs is a lot. It's a real lot. All right. Any questions? Dan, -80 shouldn't work for human cells. You can try it, but normally people, I think, do... What's the temperature? Let me look. I think it's like -120 or something like that people normally do. Let's see. So -135, I think. Yeah. So I don't know. I've not tried to store them in -80. -80 is usually out of the range of most, biohackers and labs and stuff like that. You can try it out and get back to us. That'd be great. What I do is, once you get your cells and once you are getting the hang of things, split one of your bottles of cells and aliquot some of it out, and just stick it in the freezer, and then like every 3 days or something, take it out and try to start a culture with it. And after that, you can check and see like how long the cells last. I tested, from like 4 Celsius to like minus 20 Celsius, and it didn't really work. So who knows? Who knows? It's worth a shot to try it out. All right. So next, okay, so we got our flask of cells. So once you receive your cells, keep them at room temperature, right? So try to keep it in a place where the temperature doesn't fluctuate a lot, or keep it at 37 C. Set up your heat mat. The word aliquot just means like separate into portions. So like if I have 1 ml and I aliquot it, usually that means I'm separating it into like 4 times 250 microliters, right? So aliquot just means separate into portions. It's not, it's not so minus 80, it's not, minus 80 freezers are usually huge, so it's not really the cost that's the problem. It's the transportation, because that's going to cost way more, because they usually weigh like 1,000 pounds or something, and then they run off of 220 volts. So if you don't have a 220 volt outlet where you are, and then they suck electricity like a ton, so you're going to be paying out the ass in electricity. There's rare cases where you need minus 80 Celsius freezers. So we at the Odin do all of our stuff at minus 20, and it works just fine. In fact, I think we keep it more like a little under minus 20, because some stuff freezes that we actually don't want to freeze. All right. So you got your flask. And you receive it. So the first thing you're going to do is dump off a bunch of the media. So you just have a little media left, right? Try to be sterile. Make sure when you dump off the media that you're wearing gloves and you wash everything in isopropyl alcohol, right? And then the next thing you're going to want to do is look at the cells. So you got your microscope. It should have a USB camera. Plug it in, and then download the program AMscope. You should be able to find AMscope online. Download it, install it. AMscope. Now when AMscope comes up, it should have MD300. It should list your camera up here. Click on it, and we should get a picture like this. Should start to color acclimate. Now this camera, obviously you can tell I modified this base because the base isn't big enough for 12 well plates. You can modify it or you cannot modify it. It's up to you. Now these things down here are the objectives of the camera. So you have a 4x, a 10x, and a 20x, I think. So the shorter one is the 4x, the biggest one is the 20x. So generally when you're looking at stuff, start out on the 4x and then work your way up to the 10 or the 20x, right? This thing right here turns on the light. The camera works by batteries. Now this is an inverted microscope, so that means that you're actually looking through the bottom. Now this is important because the cells are growing on the bottom. So if you have a normal microscope that looks from the top, it won't work, unfortunately. You need a microscope that is inverted, right? So put your cells on there, and then what you want to do is you want to use these knobs to adjust the focus. And you can see on here it's already focused pretty well, and you can see all the cells, right? You can see all the cells on here. Now let's zoom in. So this is the 4x. So now we're going to zoom into the 10x. Now it takes a while. It auto exposes. Okay, we can't see anything, so let's adjust the focus using the knob on the side of the camera. And there we go. We're starting to see some human cells. These are really confluent. So the word confluent means that the density of the cells is really high. So you can see that the density of these cells is really high. Sometimes it's difficult to see the outline of the cells because they're all just kind of smushed together. But the density of these cells is really high. Now, one thing, if you're having difficulty seeing the cells, one thing you can do is you can adjust the analog gain. It makes things brighter. So it starts to brighten it up. If you want to try to adjust the exposure time and stuff like that, you can also do that. Now there's a lot of different things that you can adjust. If you want to try to get the edges more, you can start to adjust the contrast of the cells. Of the microscope. Right? And what we got here is, the cells. It's really great. You can take a picture. If you click on snap or record, you can take a picture or look at video. But yeah, so normally human cells in an image like this, they're kind of hard to see the outline, but this is what confluent cells will look like. Healthy confluent cells. They just look like a bunch of oblong, kind of random ass shapes. Well, not random, but they're organic shapes. Let's see if we can bring out the cells more. Bump up that contrast, which is not doing much. Everything you see is cells. There is no junk in here. That's the thing. Let's zoom in even closer. So what we'll do is we'll go up to 20x. Let's increase the analog gain. Okay, so there we go. Here's a really good picture of what the cells look like. Right? So this is 20x. So here's the cells. This is all cells just growing on top of each other massively because this culture is really overgrown. So sorry about that. But all those things you're seeing are cells. They are all cells. All of it. So the cells are oblong shaped. So those circular things, those are more stressed out cells. But these human cells, they should look like oblong shapes with points. They should look like rhomboids. They should look like, different things. But yeah. At minus 20, you'll get used to looking at them. But, yeah, these are the cells, right? That's what healthy cells... Yeah, the circular cells are dead or stress cells. So if your culture is a lot of circular cells, something's not right. When you receive your culture, you might have a lot of circular cells. That's okay. Don't worry too much about it. Put them at the proper temperature, and they should start to feel better in a few days, right? So once you receive your cells, look at them under the microscope. Check them out. Make sure they're still there. Make sure they're attached, right? A way to tell if they're not attached is to go to like the 4x on the microscope. So turn down the gain and swish your flask around. So when you can see the stuff that's moving, that's all the unattached stuff. And if you see a lot of stuff moving, that means that you have a lot of dead cells, right? So hopefully you don't see a lot of stuff moving. You just see a little bit of stuff moving, and that's okay, right? Okay, So the first thing you're going to do when you receive your cells is you're going to put them in a new container, right? Really sterile, just in case, so that if anything goes wrong, you have extra cells. So we sent you some of these. These are T25 flasks. That's what they call them. That just means 25 centimeters. The surface is 25 centimeters squared. So the first thing I'm going to do is I'm going to ethanol my gloves again. Keep it nice and sterile. Now, you should receive tubes of media like this. Now, these T25 flasks, to cover the bottom, you need about 5 ml of media. Okay? So what you're going to do, you're going to take your syringe, your sterile syringe, and you're going to take your filter, put it on the bottom of the syringe, pull out the top of the syringe. Now, it has gradations. These are in mLs. So what you're going to do is you're going to fill it up to the 5 ml point. I went a little more. That's okay. I went to like 6. And then you're going to sterile filter the media into the flask. Right? Try not to keep it open long. Try to keep it covered. Filter that media into the flask. Close it up. And there we go. It covers the bottom of it. So now we got sterile media in here. Now the next thing we're going to need to do is we are going to add some cells to the flask. We're not going to split the cells at first. Splitting the cells is a little more complicated, and you want to make some backup cells before you split them. So after you receive your cells this week, just do this, and then next week we're going to get into splitting cells and the hands-on process of that. So what I'm going to do is I'm going to take some ethanol and rub it all over my pipette. Keep my pipette nice and clean. Make sure it's all good. Now make sure you use your 1 ml, 1000 microliter pipette for this. Now, next thing you're going to do is take a pipette tip. All right, let's try to get this set up so we can look at the workspace and not me anymore. So you're going to take a pipette tip. Now don't make sure it doesn't touch anything. What you're going to do then is open up the flask you received, just very briefly, and you're going to scrape the tip against the back of the flask, and you're going to get some of the cells. Suck up some of the liquid, squirt it down, just scrape a little bit on the flask to dislodge some of the cells. You should start to see some floating cell stuff in there. Now, make sure your pipette is set to around 500 microliters. Let's get it 500. You're going to suck up some of those cells, 500 microliters worth. Make sure you close this. Don't keep it open for too long. And then you're going to put the 500 microliters of cells into your new flask. And what this will do is it'll hopefully seed this new culture with cells. So what we can do is we can look at it on here. I don't know why my screen hasn't changed. Let's start up AMScope again. Now, in this case, what we should see, right, is we should see lots of floating cell clumps. And that's what we're seeing. You see these big clumpy things? We're right on some bubbles. So we see a lot of floating cells and no attached cells, right? So all the cells we see are moving around because they're floating in the liquid that we just added. So you should be able to see cells in the microscope, right? So use the the adjustment knob to focus, and you should be able to see the cells floating around on here, moving around. Now, in about 24 to 48 hours, they should... Oh, what happened? Press the wrong button. So in about 24 to 48 hours, they should start to attach. So don't move them too much. Try not to look at them more than once a day, because when you move them around a lot, when you change their temperature a lot, it's just not good for the cells. So try not to move them around more than once a day, right? So this is your first goal once you receive your cells. The first step is to take the flask you received, dump off some of the media, scrape off some of the cells, and reseed it into a new flask. This will be our backup flask in case anything goes wrong, anything gets contaminated, or anything like that. So the flask we scraped the cells off of, you can put it back, on the heat mat at near 37 degrees. That should be good, and just keep it there. It'll be able to survive the whole week. If you see the media color starting to change, feel free to change the media. And the way you change the media is you just dump out the old media. And these small flasks, they hold about 3 mLs, right? So just add 3 mLs, dump out the old media, add 3 mLs. Now, when you dump out the cells, try to dump them into bleach. So generally what I do is I keep a little Tupperware container that has some bleach in it. And whenever I discard anything from the flask, I discard it in there. That'll just make sure the cells get killed. And then you could discard them down the toilet or whatever. But yeah, discard the cells into bleach so it kills them. Keeps the regulators and everybody else happy, so they don't hate us. But yeah, this is what you should do and receive your first week. Store your media in the fridge. It precipitates if you store it in the freezer. So don't store the L15 media in the freezer, because some of the salts and stuff will start to precipitate out. Yeah. Any questions about all the stuff we went over today for the class? Any questions? Yeah, so be kind to your cells. Treat them with respect, and they will be good to you. So what we're going to do is we're going to grow up cells the first 2 weeks. We're going to learn how to split them and passage them. And yeah, Jimmy, bring the refrigerated media to room temperature or 37 before you use it. Keeps the cells happy, right? Warm up the media whenever you're taking it out of the fridge to use it. Yeah, keep the cells happy. They love to be happy. Always sterilize the media before you use it. To make sure that you don't get any contamination. Always wear gloves. Use isopropyl alcohol to keep everything clean. But yeah, so the first 2 weeks we're going to grow, and the second 2 weeks we're going to learn to do genetic engineering of these cells. So it'll be really fun. If your cells die or anything like that, or anything bad happens, then let us know and we'll send you more. Ronnie, yes, if you refrigerate the cells, they will most assuredly die. Do not refrigerate any cells once you receive them. Keep them at room temperature if you have to. Are these cells reliable for testing endotoxicity? Not really. Not in the way you're thinking about it. I've used like very poor, shitty preps, plasmid preps, and they're not really good. They don't die really easy from endotoxins, so it's not a good thing. I would recommend, if you want to test for endotoxicity, there are inexpensive kits that use horseshoe crab blood that work well. A protocol to make iPSCs, induced pluripotent stem cells. Yeah, that's something that we're working on. Basically, there's a plasmid that has the 4 factors that you can use to make induced pluripotent stem cells. That's something we're interested in, but we're just trying to get this going. So soon, that's something we're interested in. What genetic engineering tasks are planned for this class? What we're going to do is we're going to put in a plasmid that contains the follistatin gene and an antibiotic resistance marker. So basically, we're going to just grow up cells that don't die from antibiotics. And that's going to be the goal. The bioluminescent, can I spray that into pumpkins and it'll glow as long as there's pumpkin media? I've never tried that. Usually what you can just do is you can grow up a big bottle of the bioluminescent E. coli and stick it in the pumpkin and shake it up and stick it in there. Maybe run an air pump behind it so it bubbles and keeps it glowing. But yeah, that's what I would suggest. David Ishii, I think on his Patreon, has a protocol and stuff on how to do it. So support David Ishii's Patreon. You can find him. Just search him on the internet, David Ishii Patreon, and you can find further information on how to make bioluminescent pumpkins using engineered bacteria. All right. Any more questions about the class? Any more questions about the class? If there are no more questions about the class, then I am going to take off. If you have questions and you're taking the class, post them in the Google Classroom, or message me on Facebook or something. Get a hold of me, email me, and I'll be there to help. You should receive your stuff soon. If you just ordered it recently, give us some time. We're trying to get it to you as best we can. So be patient. We're doing our best to get everybody these cells. But otherwise, same time next week, Monday, 4 p.m., we'll go into the second part, how to split and passage cells. Thanks, everybody, for coming. Talk to you soon.